A very good morning to all the delegates and the one who are watching us on YouTube. Warm greetings from RBVRR Women's College of Pharmacy. I welcome you all for the second day of international webinar on concept of QbD in formulation development. Today I invite Dr. Sudha Parimala, HOD of Pharmaceutical Cognizy, to chair today's session. I welcome you, ma'am.
Thank you, Anusri. A very good morning to all of you. Welcome to the second day of webinar series on concept of QBD in formulation development. Today, I am fortunate enough to introduce the speaker of morning session, Dr. Subramanian Nateshan. Dr. Subramaniam Nateshan did his BPharm from MGR Medical University and MPharm from Anna University Chidambaram and PhD from Jadavpur University, Kolkata. After which he went for postdoc to the School of Pharmacy, University of Missouri, Chennai, USA, for which he got White House Fellowship from DSD. Sir has got a vast experience of 22 years of teaching and research. He worked earlier as a lead in charge University College of Engineering, Aryalur, and as a zonal coordinator in Anna University in the examination system. At present, he is working as a professor in the Department of Pharmaceutics, Naipur, Kolkata. Sir received more than 1 crore funding from various funding agencies like CST and DBT and ICMR, and also received the Best Innovation as well as the Young Scientist Award from the government of Tamil Nadu. Sir has 75 publications to be studied in the peer-reviewed journals and 15 book chapters to be studied. He also has 2 US patents and 1 PCT patent and 1 Indian patent. Now, on behalf of RBC Women's College of Pharmacy, welcome sir to begin with his session. Welcome, sir.
Thank you, madam. Thank you for your introduction. Hope you can able to see my slides.
Yes, sir.
Yeah. Now, first of all, I like to thank the opportunity provided by that BVRR Women's College for me to meet that people during this pandemic situation through this webinar. So today I'm going to talk about some of an design of expert concept which has been adopted during development of formulation at our lab. Today, the trend is that we need to have a quality product. The product need to be developed and manufactured to obtain or to attain that required quality. So to achieve this, we will be imposing or we'll be adopting various control measures. Earlier, we used to have that once the product is made, we'll have that quality control check. So once it's passed that required quality control testers, then the product has been released into that market. And later on, the people thought that, okay, that a lot of batches are failing during that manufacturing. So we need to have some in-process control. During that manufacturing itself, we need to sync or identify some of the parameters so that output product will have or attain that required qualities. Then that came into that picture. Then the people thought that, okay, that still it has some problem. We need to validate that machine, equipment, process. So the validation came into that picture. We have adopted it and we have developed the product and manufactured, attained that quality. That's what happens. And during that 2000, the people has thought still which need to be improved, as well as the time spent on that evaluation of the proposal which reached by that or reviewed by that FDAs are also becoming an huge amount. Then they were also receiving some of that applications which is related to changing of an minor changes happening in that process. As I know that existing procedure, if any changes which is other than approved by that FDA need to be get again approval by that FDA for that marketing of the product. That is the process. Now, the one end, that FDA is continuously receiving a newer application where that product dossier for that evaluation. Again, that changes which has been made it in that product is also receiving it by that FDA. So that makes them a lot of difficulties to analyze it. Again, there are some critical factors which need to be identified or which need to be directly correlated with a product quality that also need to be identified, that came into that pictures. So that several guidelines has been brought out by that ICH, International Conference on Harmonization on Product Development, particularly that Q8, Q9, Q10, and 11, which made that basis for that quality-based design. Now, the people has thought, if we are doing and quality-based design, so that you will be getting a better product. Now, this quality-based design need to be adopted during that initial stage itself. It has to be starts from and formulation development stages. By keeping what is required at that end of that product, or what is required qualities at on that product. By keeping that quality-based design concept has been introduced. Now the lot of industries are adopting it. When you are submitting a dossier for an FDA, that process need to be clearly mentioned. Whenever we discuss about an QbD, the people will always say that, some of the people will always say that, sir, we are also doing it. When we inquired a little bit more, they were just doing a design of an experiment. The doing of a design of experiment, it's not an QbD. That's what my slide shows. The first thing, QbD is more than just DOE or a design of experiments, whatever. Now, I'll go with a little bit introduction, but I know that last two talks you might be heard about it. But in order to go further, I'll give some inputs over here. According to an ICH Q8 or that QbD defined as a systematic approach to a product development that begins with predefined objectives and emphasize the product and process understanding and process control based on sound science and quality risk management. Whereas the design of experiment mentioned as the systematic method to determine the relationship between factors affecting a process and that output of that process, which clearly gives an information about an cause and effect relationship. Now, before going into that in detail, I put up a simple example. So every one of us will like to have a morning breakfast, particularly if we have a puri, we'll be very happy. But some of us as a diabetic patient, we like to have a chapatis at a night, or we like to have a chola puri at a bigger size, having with an sabjis. Now, if you think that raw material which is used for the both that making of an chapatis and puris are similar. Only the difference is the process which makes a different product and different qualities. I think our friends knows very well, while making a dough, we need to take that wheat flour, we'll mix it with a small amount of an oil, add up and water. And this need to be mixed and kept it for some time. Then we'll make it into one slices that can be put it into tawa to get it and chapati. If you fry it, you'll get it as a puri. That's very simple logic. Now, where this quality-based design or the design of experiments comes into that picture. Now, the quality of that material or that food, what we are taking it at that end, will depends upon that source. What is this, that wheat flour, that quality of an wheat flour. Now we have different kinds, fiber rich, gluten free, or we can make it as a homemade. We can also see that advertisement which is giving up even and put it on tawa, you will be getting and wheat flour is buffet like puri. That's happening. That is a real time examples. Now it clearly indicates that input material or that wheat flour has an impact on your end product. That's an example. Now, if we come into that process, everybody makes that chapati. Everybody makes. Now, the question is, how many of our product is liked by that people or people interest to have it? So we also make it at home. But I also tried sometimes to make that chapatis, but at last I got an very hard one, which need to be soaked in an sabji or an sambar before cooking it. That is my process. Where it is gone wrong, that is that key. The amount of water which is added to that flour, that amount of oil has been incorporated, and that time for which we are allowed to soak it, that process made the difference in that output. So, from an initial, every steps need to be correlated and made it carefully so that the output, whether it is an puri or an chapatis, which can be takenable or eatable by the people. So, the entire process need to be adopted quality-based design. We are adopting the techniques on our everyday life. On each and every activities we do it. That is adopted unknowingly. We are doing this. Only thing, whether we have identified the criticality on that process or in the process parameters that need to be discussed and identified. Now, if I'm coming into that DOE, I've made it clear that is a relationship between the factors affecting that process and output of that process. It's very clearly mentioned how we are going to determine that relationship. So how much time I can put that rolled tow mass or that top mass in that pan, how much time I can dry it to get it and chapati. That need to be optimized. That maybe depends upon that. It may not be a time specified time, we will not be adopting on our day-to-day life. But if it is a machinized or processed, the time need to be fixed. If you are over fried or over time, if you are put it over that tawa, it will be dry, so it will be very difficult to tear off it. If you keep it too light or lesser time, it will not be properly cooked. So that time optimization need to be done. So where that DOE comes into that picture. That's at one point. Process effect, or the process directly influencing the output of that product or the quality of the product. That's one case. Again, this DOE will be useful to find it out that relationship between that components added. I'm going into that same example. Adding a fan wheat flour with water. We know that difficulties. If we added that more water, which will be difficult to roll it into one slices. If do not add that sufficient water, which will we need to apply and more pressure to roll it or made it into thinner slices, that kind of an relationship will be established by using and design of an experiments. This is the simplest concept. But when we are having more and more factors, more and more amount of an process coming in or input variables are coming in, then the finding out the relationship between that input variables and quality of the product becomes complex. So that complex nature can be studied by using a design of experts. Before going into one detail, I'll put it as an QbD. The QbD is required to develop a product which meet out that required qualities as well as provide an opportunity for continuous improvement of the product. That's what this slide shows, that development cycle wherein that requirement starts from a patient, what kind of an requirement is needed by the patient, and that make it into that product requirements. Once that product is designed or fixed, depending upon that process, we will able to arrive that manufacturing process and its developmental. Once that manufacturing process is designed, then what are the factors or that variables which will affect in that process need to be identified and it need to be analyzed. Therein, that risk assessment and analysis comes into a picture. Once that is done, that will be correlated with patient needs. We can continuously adopt this cycle and improve that product quality until it meet out that requirement of that patients. Now, if I'm coming into this once again into that wheel of a development cycle, the patient needs will clearly indicate what is that requirement to be there, or that what is a target for that product which need to be developed, which simply called as a target product profile. For every product, now we have a target. Say for an example, if we take it as a conventional tablet, we know that which has to be disintegrated within 15 minutes and 85% of the drug need to be released within 30 minutes. That is a target profile. It should have suitable harness which withstand during the transport, and it should have that suitable stability during the storage period or a minimum prescribed period of 3 years or 1 year, depending upon that product. Now, this called as a target product profile. That's already there. Now, the question comes, when the new product need to be developed, what kind of a product we are going to develop? Whether it is a tablet, capsules, the ointment, or a novel drug delivery systems, that comes into a picture. Once that is decided, the product, okay, say for an example, I'll be going to make a tablet or a conventional tablet. Then the product requirements comes. If it is a tablet, what is a quality target profiles? That comes into that picture. Once that's done, then we need to identify what are that inputs need to be given related to manufacturing process, where the two things are coming into that picture. One is an raw material inputs, another one is an unit operations. That means the quality of that raw material and the process what we implement to get that product. Once that is decided, the way the critical process parameters, critical quality attributes of an material attributes, that will gives and various parameters. That influence of these parameters need to be assessed properly. Then that relationship can be established once again. Then that cycle can be continued for that product developments, continuous product development. So, in that sense, we can put it as an design of an experiment is an element of an QbD strategies, but not only the part of it. Again, we can say that that QbD vision, according to that ICH guidelines, we can say that start with an end in mind. That means whatever that product you want to get it a quality product, keep it into that mind, design your developmental program accordingly, which has some various essential components. This has been discussed widely by the two speakers earlier. I think that anyway, here you already came across the process. One is an fix that quality target product profile, then determine that critical quality attributes and assess that risk involved, where we need to make a link between an material attributes as well as an process parameters to have to identify that quality attributes, critical quality attributes. Then based on that risk assessment, develop your own design space or what is an available design space. After identifying the design space, implement it or adopt and control strategies where you design it and implement proper control strategies to work it within the design phase and achieve that product profile or quality profiles, so that we'll able to continually improve that product, or we can able to manage the product life cycle. Now, I'll put it in simple example how this QbD approach for the development of topical semi-solid products can be put it over here. Till now, we have discussed wherein we need to identify that what is the process is involved, what is the material is involved, which will be called as both the cases will have an input variables that will affect that output variables. Wherein we already discussed, the people have discussed, which is called as some critical process parameters and critical material attributes. If you take some solid dosage forms, what could be that our product profile is a topical semi-solid product which should have some suitable spreadability and should not irritate during that application, and we should have required drug content. That is a quality target product profile. Now, in order to get, we have that critical process parameters and material attribute. If I think about that material attribute, several parameter attributes like API purities, API quality grade, excipients and its quality, and physicochemical properties of the drug and excipients will influence or that attributes. When you are coming into that process, say for an example, semi-solid, we might use melting of that components and stirring or mixing of components. So the temperature control, type of that mixture used for that mixing process, what is that mixing speed and time, and homogenization, how that homogenization of that different excipients and drug has been done, milling to get it and required size of an API or an excipients and order of an addition. This will comes under these categories that will decide. Based on that, you will be getting a critical quality attributes. What are an attributes? That is an particle or a global size, polymorphism, pH, rheological properties, evaporation of volatile materials, and what is a suitable container and closure systems we are going to use it. whether these quality attributes are able to meet out that specified limits given in official compendium or an official pharmacopoeia and users requirement. Here that point is very clear. I mentioned it earlier. If you are talking about a semi-solid product, it should have suitable spreadability, non-irritant. It seems to be very easier to understand. When you are having a solid disposed in an ointments or a cream, if the particle size of that API is not properly controlled, then which will produce an irritation. If the rheological properties or the viscosity of this product is not properly addressed, you will be having a problem in spreadability or in an applications. So these quality attributes will be defined. That will be depends upon your process and material inputs. Once this has been done, then post-manufacturing controls need to be studied, particularly stability and quality by testing that control testers and compliance to an official requirements and find it out as an irritancy of that product on application of an skin surface of an animals and human establishes that product, whether if it is retained and occlusive properties, it has an occlusive, non-occlusive properties, that kind of studies can be established at an end. This is one of an approach which can be adopted for that QbD. Now, my talk will be more hereafter will comes into that area of an design space only. I'm not going to discuss much about that other spaces. That's what my subtitle sound, use of an design of experiments for developing and systems, which is a nanoscale examples. I'll be going to give it on forthcoming slides. Before that, I'd like to give some information about an design space. Now, you just see here that is a picture wherein you can able to see that any product development which has an opportunity or an operating ranges, that is called normal operating ranges or the process parameters. Now, once if it is there, our knowledge is wider. Our knowledge is wider. Say for an example, I take it as the amount of surfactant to be used in our system, or an use of an diluents or an glidant in our products. The approved range is varies. Approved range is varies. Say for an example, if I keep it at 5 to 10%, or an up to 10% is approved, that will comes into an design space. Or 10%, I've put it as an approved, we can keep it as a knowledge space. Now, I'll be making a formulation with a 3%. That will comes into that operating range. If I expand up to 5%, that will not affect or that will not have an great influences on that product qualities. That range will comes into an design space. Now, our requirement, we need to come up with an design space. Other than that normal operating process or that range, we need to come up with a certain space where later, if we make it as any changes for which you need not to get it as an approval by an FDA. That's what that guidance is very clearly given. If any changes made in that design space does not require an regulatory approvals. So just a minute. Hello. Yeah, I'll call you after 12 o'clock. I'm in meeting. So the design space, nothing but established range of and process parameters that has been demonstrated to provide assurance of and qualities, where we need to identify that high and medium risk area by proper risk assessment. We know that there is some flexibility given by that regulatory authorities, wherein the design space is not generally considered as a change of an approval, approved changes for the process parameters on that formulation attributes. That's what I was discussing. So the changes within this space would not entitle for that regulatory reporting requirements. So we need to identify or study properly the design space, which is a quantitative understanding of how API, formulation, packaging, and process parameters affect that drug product quality attributes. Now, if we come into that, how this product will maintain its robustness even after changing an inputs or minor changes on that inputs, or what are that changes in that input, how that quality of the product is getting varied. Now, one such example is shown over here. What is that? We know that two kind of an inputs. One is an material inputs, another one is an process inputs, where if that inputs, material inputs and process input will be processed, that is a process step. Once that will gives us a product where you will be having an variability in that product quality. Now, I take it as this example. In this picture, we can say that particle size. Particle size. There is two lines. One is indicating by a green, which will gives us a product having a narrow particle size. If I make it as a red color one, that is having the product having a wide range of particles. I can say that from an nanometer to micron level, whereas that yellow is green is having an particle size range of 100 to 300. For an example, I'm saying. Now, if you are coming after that, the particle size will be getting increased. Going down, the particle size is getting decreases. Now, your process inputs will determine how these particle sizes are of your product. If your process is proper, process parameter is optimized, always you will be getting at as an green color one, that is a product having a narrow particle size. If your product is or the process is not optimized or the parameters are not optimized, then this will give some variation in your particle size. Now, if there is any little bit changes, that also need to be identified in the process parameters. Now the question has come, only the process parameters will influence us or that raw materials will also influence us. It is very clearly shown that not only the process, that raw material will also influence us. If we are having a material which is very hard to break it, or that API, what you are giving an inputs, if you are developing a process which has that API particle size of 5 microns, if you are later on, if you are changing that API into a 10 micron, then your particle size of your end product will not be similar. So that input variable will makes the difference in your product qualities. So the robustness of this product need to be assured by doing a proper process, wherein the process also should have an robustness, which is called as an process robustness, wherein the ability of the process to tolerate the variability of material changes of the process and equipment without having negative impact on the product. That is called process robustness. That's what I given an example. Take it as the same example. I'll be giving a particle size of an API having a different sizes, and 5 and 10. If that is there, there might be a huge range difference in the particle size. That process may not be optimized, or my process may not be robust. But I can able to say that, or I can able to make a product. See, for same example, I'll be having a 5 micron and 7.5. My process is a robust, which will gives the same average particle size or a narrow particle distribution. When I'm having an input material having of 5% and 7.5%, that is called process robustness. So when you are using this QbD, we need to ensure both the product robustness and process robustness. That process robustness can be obtained by using a design space. This is an area we need to concentrate. Whatever that input parameters and both material as well as the process that need to be done, based on that product output that can be monitored, you can adopt and process analytical technologies or process control technologies. Based on that, you can alter that input variables, get it and required product qualities. Now, in order to get it a process robustness or a product robustness, what is an area or that tools can be used? That is an design of experts, where we'll use different kinds of designs, whether that back factorial design or anything. You can keep it a mixer design or an response surface design or response surface methodology, any kind of design that can be used to identify that interaction between variables. Yeah, I'll say. And we can also plan that to get it and required output, we should have that expertise to maximize their return. After that, we need to have a proper analytical modeling or a mathematical modeling to analyze the design space or to make an output. Now, earlier the people were using an an artificial neural network. Now there are a lot of technologies has come up. Now the people are going with what is called artificial intelligence is also coming into that place. By using that, lot of analytical modeling nowadays done that can be used for that design space. Now I'll come into that, what kind of different types of experiments can be used for obtaining this or to get this and process robustness by using of a design of an experiments. Either experiments can be carried out by a trial and error method, wherein will after getting a successful result will have and proper design space. If you are making one trial, getting a specific result, then which will not give that a proper methodology or the design space or that robustness that meet that quality-based design submission. Where there should be some of an factors or an requirement we should have to arrive or to meet out this area or that design space, for which we can go with one factor at a time. That may not be an insufficient strategies compared to an design of an experiments where we might miss an optimal design space. So that we'll go with a screening with a design of an experiment by having a loop factor designs to identify that important factors at a development process. Once that is known, then we can redefine or refine the DOE design of an experiment by fitting a mathematical model. And once that is done, we can optimize that product by using an optimizing designing of an experiments. So how it can be applied? Like trial and error, we can have that specification that gives set of levels, as well as will screening DOE will done with 5 to 10 factors at 2 levels to identify that factors, then refine the design of an experiment and optimize that experiments with 2 to 3 factors, often having on variables as well as levels at 3 to 5. That will give us a lot of information or required information about interaction between these excipient or a contribution of these excipients on quality attributes. Once that is decided or obtained, we can run that confirmational studies or a confirmation run to verify the design space whether which will gives and required outputs. These confirmational runs can be carried out and any area or the points can be chosen from any area of a design space. Once that confirmation is done, then we can go for a submission of a product development dossier. Now, how this need to be applied in a real practice. So initially we need to determine the responses, both input and output, and choose the factors and set that level for individual factors how to what extent it is critical, whether it is at low, medium, high. Accordingly, the levels can be kept it, and uses and different software packages to design and analyze your data and run that experiment, get the data, analyze it, refine it, and optimizes that experiments. Now, I'd like to give some information about that mixed design. I think a lot of information or everybody's talked about and factorial design, the box cost design, surface response methodology. A lot of things has been discussed last 2 days. I'll put across another concept. Some of you might be knowing it, and mixed design. Now, these mixed designs are used when that response changes as functions of relative proportions of that components. That means when you are changing that ratio of that components will make us that changes in quality of a product, which will not depends upon that quantity of that materials used. Clear? Whenever we take very simple concept, having a rice, cooking of a rice in cooker, we will optimize depending upon that rice. I think everybody knows that. Generally, we'll keep it as a for 1 cup of rice, 2 cup of water. In general, I'm talking about it. So, if I raise that water level, okay, the ratio is raised from 1:2 into 1:2.5, then what will happen? The product will, or the rice will become sticky. If it is 3 or at 3.5 instead of rice, I'll get it as a sun pongal. So that kind of a relationship will be better given by that mixer design, not by that factorial designs. So all the component must have that same units of a measurement, and each run has to have some of that total. Some of that total. I'll take it as a same example. Making of a rice. That is anyway, I'll come into another example. Making of a same rice. The 2 cup of water and 1 cup of rice. So totally 3 cups, that is fixed. So if that ratio is changing within that by fixing and 3 cups, if I make that changes within this ratio, instead of 1:1, okay, or 1:2, then that will give us a variation in that output of the product. Now, I'll take it as some of examples is given by that design expert software teaching module. These slides, some of the slides has been taken from them. I acknowledge them for these sources. Now, what is a mixer designs? First, I'll put it as an example of a packing of a cake where we have the 2 parameters. One is time, another one is temperature. As you know that cake need to be packed in an oven by setting of a time and temperatures. Now we'll have the 2 parameters, input parameters. One is on time. It can be varied between 20 to 30 minutes, and temperature from 300 to 450 Fahrenheit. So by keeping these as input variables or an input responses, that output will be measured in terms of an moisture content present in the cake. So both the factors can be set independently. Both the factors can be set as an independently. Either I can make 25 minutes, the temperature value, if I keep it at 25 minutes, the temperature values can be set it at any time. That is an 300 to 450, anything, any values I can set it. So that will give an response surface methodology, or an this analysis can be done by any kind of an factorial designs. So one such experiment has been given. You just see it over here. Factor 1, varying the temperature from 22 and 30, or that temperature can be varied by from 350 into 450. Anything can be varied accordingly. The moisture content in cake are measured, that relationship established, but it's need not to give us a common relation. There is an individual thing at 450, that at 20 minutes will get it as a moisture of this. If I have that 450 at 24 minutes, here that, so that can be varied. The difference can also be varied. Now, you just consider an example to making a pizza wherein you want to keep that cheese so that pizza will have suitable cheese content. While taking it, it will melt smoothly, and while pulling up, which will give us an impression that the cheese is there, which will feels like elastic in nature. In order to have, we'll have that different mixture of cheeses or blend of cheese such as an mozzarella or provolone and white cheddar. So for making this three thing, that composition or the total quantity is fixed as 6 ounces. Now take it as an example point over here, so that all the three has to be mixed in a particular ratio or the proportion meet out that total of 6. If your experiment having this kind of a constraint, then you need to go for a mixer design. You will not able to go for a factorial designs or in response or some methodologies. Now, within this, we can arrive that this product maybe varies from 0 to 6 ounces. So, based on this, that experiments can be designed by keeping a 0 at first one, 3 and 3, or by varying 0, 0, or 0, 6 and 0 in other areas. We can have a 16 number of an experiment, but wherein all that runs will have 6 ounces of cheese at the end. This is a classical example where that mixer designs can be put it into that picture. Now, what is the difference between an factorial designs and response surface method or some, and come into that mixer design? Now, in factorial designs, we'll always consider about the design space as a rectangular, wherein we will not discuss about a fourth product. So, whereas I'm not going into deep into that anyway, wherein in that mixer design, we will be the design space will be considered as an triangles. So we will be able to get it as a triangle over here. So that in this case, earlier case example, you just come over here, that triangle representing each end of the triangle is representing 6 ounces of any particular cheese. If you take it as an A, mozzarella, it is an 6. Here, that white cheddar is 6. Here, that provolone is an 6. If I am moving on this line, so I'll have this is a point, say for an example, and 3 ounces of an A and 3 ounces of an C, or a mozzarella and cheddar. If I'm coming over here, I'll have an provolone and mozzarella. If I take any points inside of this diagram or a triangle which represent all three things, that is the concept I like to put it over here. Any point which within this design space where or this triangle will give a combination of an 3 that cheeses that will decide that quality of the product. Now, what I learned, so I think this is a difference between an factorial and an mixer design, where that mixer or an unit is fixed, wherein the ratio will assess an influences. In factorial design, that's an individual parameters can be varied. Now, if you take it as an any point over here, okay, so if I keep it as an 50% of an A, then the other two will be varied accordingly. It can be fixed only at an within that 50. So say for an example, if I take this and point over here, having an 50% of an A, 25% of an B and C. If I come over here, that will give 75% of an A and 12.5 of and 12.5 of an E and C. Like that. That will changes within that 100%. Or if that range is fixed 0 to 1, within that will be changes. Now, I already mentioned it, that design space for that mixer design is a triangle, so that generally this design space or the two categories of an mixer designs can be done. One is called as an simplex design based designs, where that canned or that triangle based things can be done, or a computer optimal computer generated design that can be used, which can offer a lot of flexibility where the material is having an variabilities. Now, I put it as a simplex design, where you already have mentioned it. These designs are straightforward. We have that differences ranging from 0 to 100%. We can have it like what in earlier cases we have seen A, B, C, that each point represent 100%. All the component must be in that same range. So that simplest design, which looks like this one, wherein you have two factors or three variables of an A, B, and C. If you are having more variables, then that will become an tetrahedral. So that will also put it over here, where we can also have that simplex design with an augmented. That means we'll have a repetition points or constraints. Then we'll go for more points over here. Okay, so centroid lattice design, simplex design. That means where you will have a repetition points at both that levels, all that levels. So that number of runs will be get little bit increased, but your product is more optimized. So that simplex designs are restrictive and limited purposes. When you are having ranges from 0 to 100, which will not able to give 100% output, since whatever that we use it, we'll use it with a restricted range. In such cases, that optimal designs are most useful. or when you are having a constraint, that optimal designs are more useful where and single components like A, B, C, which can be varied from a 0 to 20% or a 0 to 50% or 0 to 50% for B and C. So if this kind of constraints are there, then you need to come out over here. Now, if I'm making a microemulsion or a nanoemulsion system where I will have an oil, A of an oil, B of an surfactant, C of an water. Now, within this surfactant and oil, I can fix it that ratio A and B should be not greater than that 10%. Then which will become as a multi-component. Again, I can put it on further restriction. Whatever that restriction, which leads into that final conclusion that A, B, and C, and addition within that 100%. Now, you might be clear when you are coming this kind of a constrained environment, the computer generated the optimal mixer designs can be used in a better way. So that's, you just see that levels are fixed, A and B and C, from a 0 to 20, 50 and 50. Accordingly, the constraints can also be put it over there where we can come up with an required design space. Now, you look about these computer generator systems. Now, if you see over here, I think one slide is missing. Anyway, if I look about that triangle, earlier triangle, if we are seeing over here, the points over here, the triangle, if we consider that after obtaining or putting this constraint, that will not really fit it into an outside of the triangle, that will comes into this particular area. When I go very particular, only this white area, which this constrained area will be called as a design space, where that experimental runs need to be done, where you will have that different points. These points can be utilized for designing of an experiments. So with these approaches, we can also design that different mixer designs that can be analyzed and mathematically optimized to get it and required product qualities. So with this information, I like to go with some of our experiment which has been done at our laboratory by giving some inputs about both self-microemulsifying drug delivery systems as well as nanomicers, which has been developed at our lab. Now, as we know that self-microemulsifying drug delivery system is a mixture of a drug, oil, and surfactants, along with or without co-solvents, which forms and fine oil in water or in water in oil microemulsion upon dilution with water, or after administration, after oral administration. Simply we can say that it is a mixture of components of oil, water, oil and surfactant when it is administered, which leads to formation of an microemulsion, whether it may be O by W or W by O, wherein the solubilization of an excipient matrix or an interface and dispersion will happen in that GIT tract due to that retropulsive repulsions occurred at our GIT tract. So this product will gives very high surface area to volume ratio as well as these microemulsions will provide a faster drug release and in reproducible manner. Again, these microemulsions are a self microemulsifying drug delivery systems are independent upon an pH physiology as well as a feed state can be made in. But even though that feed state condition has the greatest influences on this component that need to be studied and optimized. So we developed an micro simers for a drug called as an celecoxib. Everybody knows that it is a specific COX-2 inhibitor. It is an hydrophobic in nature, having a low aqueous solubility. The greatest problem with this product is available as this drug is available as long needle-shaped crystals, which shows a lot of cohesiveness, bulk density, and low bulk density and compressibility. So the earlier product is in capsule form, and most of that product is available in that market is in capsule. Very few are available in that tablets. Now, which is also how that T-half of 3 hours, Tmax of 2 hours. This product is also recommended for a treatment of post-menopausal pain. We thought that if that Tmax achieved in shorter duration, then the pain can be reduced quickly. So with this move, we have designed simers to enhance the solubility and to get it as a quicker or a faster onset of action, for which we have optimized that formulation by using an mixed design methodologies, for which we have initially identified. I think I'll be a little bit short of time. I'll go first. First, we'll identify that solubility of drugs in an oil and an surfactants. Based on that, we have chosen that 2 surfactant and oil for our formulations. Okay, wherein we have used this as a different oils and surfactants, Captex 810, that is a triglyceride, as well as a triesters of an propylene glycol, which is called as an TPG. Again, another oil we used much is an propylene glycol monocaprylate esters, MPG, or an PGA, caprylic and capric glycerides. If you see it from this, these caprylic esters, CCG, will have a lot of solubility. Again, this propylene glycol is a propylene glycol monocaprylic esters is also shown in high solubility, and diesters of propylene glycol has been shown 14.7. So these two has been tried, either can be used as a co-surfactant so that the solubility of that formulation can be enhanced. So we made it 2 in 80 as chosen as a surfactant. With that, we made it into a different ratio, 1:4 into and 4:1. Find out the solubility of drugs. Be able to get it as a maximum solubility at a 3.1 ratio, but later on it becomes flat. The same thing has been observed when we are having a ratio between a 2:20 and MPG. That is a monocaprylic esters. Now, based on that, we have made it in different pseudo ternary diagrams by using a 2:80 TPG and of a surfactant system, CCG as an oil and water at 28 degrees Celsius. This black color does not yield any microemulsion region. The other area will give us an microemulsion regions. Similarly, we have developed a system, pseudo ternary diagrams, for this system having an 20 mpg as an surfactant and CGG as an oil and water systems. Now, we'll come into our pictures in order to optimize that composition of these systems. We have used a design of experiment, particularly Simplex Lattice mixture design. We have adopted a two methodology initially in order to have better understanding of that formulation. We had only surfactant, oil surfactant mixture and surfactant. We had that percentage that design points has been made or arrived from a Simplex Lattice design that is a combination of these two, OSM and CGG, will fixed as a total fixed as an 100. You can see it from here at any point, which gives us an 100. And then response variables, we kept it as an absorbance. That means after mixing of these two in a required proposition, diluted in water, and that absorbance is measured at as well as an transparency has been measured at an particular wavelength, or that 400 nanometers. If that absorbance are less, then it is indicating that the product is transparent in nature. Similarly, the solubility has been measured. In order to get this data, we put it into an equation or establishes a mathematical model. This model fitted clearly in linear models. These parameters can be put it into different things. Either it may be a quadratic equation or a linear equation, and parabolic. Different equations can be adopted. This study provided and best fit line with the linear model. So based on that, we have developed an equations. This equation gives an point you want any solubility or insolubility of any for the particular product. What need to be the composition that can be arrived from this equation. So from that, we can also get it as a what is your design space where you can play. Similarly, we have done another methodology where we kept it as a sand. We incorporated drug as a 10%. The variation has been done between an oil surfactant mixture and with oil of an what is called as a fixed percentage of an 90. Now you can see from here any design points is a 90. Another 10% will be represented by your drug substance. Here it is essentially coxine, where we have kept it as a response variable of an T85%. That is some time taken for release of an 85% of the drug. The T85 is one of an important criteria as per that FDA to assess that dissolution properties of that conventional tablet or unconventional product. Similarly, we also kept it as another response variable as an dissolution efficiency, which will varies what could be that on depending upon that composition, how that efficiency of that product is getting altered or the dissolution is altered. So by adopting a Simplex Lattice mixture design, we could able to find it out that dissolution efficiency can vary between 78 to 92, while that T85% varies from 96 minutes to 27.6 minutes. There is some greatest variation while changing that compositions. So that has been fit it into one model that is given as an linear model. Again, it is fitted into a linear model. So we got it as an equations. Now, our aim is to get it as an lowest time for and best lowest time for T85 and better efficiency. So based on that, we arrived at compositions. We did that formulation as well as we calculated that what is that experimental result and what is the same calculated results. What is the difference between an error percentage between an experiment and calculated? That we have done during that experiment also. You can see it from here in case of an experimental value, which is coming for our optimized formulation 0.022. The calculated value is 0.021. So with it, little bit percentage error of 4.5. Like that. Whenever you develop that model, fit that equation, get the data, run that experiment, analyze your data carefully. So this need to be done clearly. So this is giving about an information about your dissolution profile of that. After that, making and formulation, we also quantify micrograph or SIMP micrograph and silicoxib has been taken. You can see that clearly needle shape. After that, we can see able to changes in that shape. So we also did this an oral bioavailability in a human volunteers. Okay, it is not possible at nowadays by an academic institute. This study has been done during 2003 and 4. So we are able to do it after obtaining an institutional human ethics committee approval, where we have compared with an marketed capsules. Our self microemulsifying drug delivery system is able to show increase in that AUC as well as a faster onset of action. The Tmax is achieved within 1.5 hours instead of an commercial capsules of 3 hours. So AUC also better in case of an CMAX. So the formulation optimized by using an Lattice design can able to give it an better outputs. Similarly, we have used and developed and self-assembled nanomicelles by using an PVP K90 and poloxamer. Hereafter I'll refer PVP and poloxamer for this grade for that delivery of artemisinin for the treatment of an AMD. I'll be just going quickly. This topical formulation, ocular topical formulation, is developed for that effective treatment of an AMD is called as an age-related macular degeneration, where your central vision is affected due to formation of new blood vessels. That new blood vessels are fragile in nature, so that will create a lot of fluid in that systems. So that will also affect or make that damage of an macula. So your vision will be affected. So for which we have taken as an artemisinin, which assesses that porous solubility. For that preliminary screening study, we have taken an PVP and poloxamer at a different ratio, dissolved in an ethanol, dried, and then we have incorporated in an artemisinin also. We'll get it as a drug-loaded particle that again disposed in a sterile water to get it as a nano dispersion. That has been visualized examined and different ratios has been studied or range has been arrived, then we made that composite. Hello? Hello? So we adopted a central composite response quadratic model design at a 2-factor 3 levels, wherein we have had an input variables of an polymer concentration, PVP, and polymeric surfactant between an 0 to 5%, polymer of an 0 to 10%, response variable of an particle size and transparency. We adopted an experimental design by varying that factors. We got an responses in terms of an particle size and an variation in the transparency. After getting an responses, we made it into a 3D picture as well as a contour plot by adopting a surface response methodology, which clearly indicates that poloxamer is positively contributing towards that or negatively contributing towards that particle size and positively contributing towards the sand transparency. Here, we show sand particle size. You can see it from here, the concentration of an poloxamer and an PVP. When you are increasing that PVP, so the particle size is getting decreases. When you are increasing that poloxamer, the particle size will be getting enhances. So that the pictures you can able to obtain by 3D picture as well as this and particle size. Similarly, we can able to see that transparency has also been represented over here. We also studied and what is a critical micellar concentration of that formulation by using a pyrene fluorescent probe, which comes around 265 and 285 for that drug incorporated and without drug. We also characterizes that the machine formulation for different parameters such as particle size, zeta potential, transparency, and we also identified the hemolytic potential and in vitro hemogenotoxicity. So we also done that in vitro drug release profile compared with a pure artemisinin suspension. Our nanomicelles are able to show better release profile compared to pure substances. Similarly, on ocular permeation against that cornea, that nanomicelles were able to provide better correlation. So we able to get it and better apparent permeability of coefficient. That developed formulation or that optimized formulation has also been after that permeation, the cornea subjected to an histopathology and what could be that possible mechanism, which clearly states that this slide, left hand side is a sand. Sir, okay, sir. Okay, sir. I'll go through it and prepare it, sir. Okay, sir. Okay, sir. Thank you, sir. Sorry, it's an important thing. Now we can able to show that radiuses and differences as well as we by using an CAM assay we are able to show that the artemisinin developed formulation is able to rupture that newly formed blood vessels. You can able to see it from here very clearly. So in a nutshell, we can say that by using of a design of experiments, we can able to optimize that formulation, which will meet out that required quality product attributes. So the take-home message is we can use often design of export to design or to arrive your design space. Based on that, you can able to meet out your record product quality and um, requirements of an QBD design. So thank you for your time. I think with that, once again, I thank that organizer for that opportunity and for the participants for listening. If somebody listens, uh, anyway, I'll take up that any of the questions if it is there.
Thank you, sir. Thank you very much for that stupendous talk. Uh, uh, for sharing your knowledge. It was an excellent talk. I think all of our students have gained much knowledge on this topic with your talk. I hope no questions are posted in the chat box.
Yes,
If,
please, ma'am.
yeah, if we come across with any questions, we will be contacting you, sir.
No questions?
Yeah, as far as I can see, uh, no questions are posted in the chat
Okay,
box.
thank you. Thank you very much.
Thank you very much. I can see how busy you are. With your busy schedule, you have accepted our invitation and shared your tremendous knowledge with us. Thank you. Thank you very much, sir.
Thank you, ma'am.
An announcement for the delegates who have registered for this webinar. The participants who have registered for participating in the oral presentation and poster presentation are requested to join to the Zoom through the meeting ID and passcode that has been sent to you on your email IDs uh, before 2 o'clock in the afternoon. So we are done for today. The participants, I repeat, the participants who have registered for presenting orals and posters can log into the Zoom meeting through the meeting ID and passcode that has been shared to you in your email IDs before 2 o'clock, 1:45 to 2. Thank you.